Why do we sleep? Why do we dream? What does this do for our minds and our bodies? Welcome to the Sleep Science Podcast, where we discuss all these issues. I'm Penny Lewis, a sleep scientist and the presenter of this show. The Sleep Science Podcast is a production of the NAPS Sleep Lab at Cardiff University. You can find out more about our guests, the podcast itself, and our research at Cardiff on the podcast webpage. My guest today is Dr Daniel Bendor from University College London. His work focuses on rodent studies of play cells in the hippocampus. We discuss the phenomena of memory replay, in which play cell activity during sleep actually mimics patterns of activity that occurred while the rodent was performing a task in wake. We talk about how this replay can be manipulated using a technique called targeted memory reactivation. We also discuss how, instead of actually triggering memory reactivation as commonly supposed, the targeted memory reactivation technique could instead simply bias the brain to replay the memory which was cued, essentially giving that memory an advantage over the others in a competition to get replayed. We go on to talk about how memory replay can reverberate back and forth between the cortex and the hippocampus, and to discuss theories about how this reverberation allows the hippocampus to train the cortex such that it can gradually knit together the various components of a memory. Throughout the episode, we also discuss rodents as a model system for studying sleep and memory, and think about what's good and bad about using them in this research compared to using human or primate models. Finally, we turn our attention to the concept of brainwashing and concerns around the idea that targeted memory reactivation could actually be used to manipulate people's memories for real. I hope you'll enjoy our conversation. So, I should start by saying welcome to the Sleep Science Podcast. Thank you very much for spending your time and agreeing to come. And it's great to have you on the show.
Thank you. Thank you for the invitation, Penny.
So Dan Bendor is a man of many talents, but I think it's fair to say most of his work involves rodents and a lot of work on play cells and the hippocampus in rodents. And the part that we're particularly interested in, of course, for this show is the work that's relevant to sleep. But this is by no means all of the work that Dan has done, of course. So he's working on a lot of other things as well. So I'm going to start by asking you about where your interest in sleep and memory came from and how you got into it.
Yeah, it's a good question. I think when I got interested in neuroscience, I was interested in the big question of consciousness, how something so mysterious can happen from a bunch of neurons. And I found that to be a very difficult or even impossible question to assess. So the next step towards that, I thought, was dreaming. What is happening in the brain when you're dreaming? It's sort of like a form of consciousness. You're having an experience. And can you actually see what is going on in the brain during that sort of activity? Of course, in animals, it's difficult to know if they're dreaming, but when they're sleeping and you see brain activity that reflects similar activity that occurred when they were awake, doing some sort of behavior, you can make a correlation that perhaps they're re-expressing the same neural activity and they're experiencing something similar to when they were awake. So to me, that was as close as I could get comfortably to looking at something like consciousness, but in a more tractable way.
And you have a very famous paper in which you actually applied targeted memory reactivation in rodents during sleep. Is that your first real sleep paper?
I can tell you how things led up to that paper.
Mm-hmm.
So actually, one of the first papers I read actually was from Matt Wilson's lab looking at REM sleep replay. So it's a phenomena of neural activity during a behavior where the animal is running around a circle, repeating when the animal is in REM sleep. So this is, in humans, REM sleep is, you see brain activity similar to the awake state, but you're paralyzed and it's associated with dreaming. So we saw the same patterns occurring, and this is the first paper I read in neuroscience. It was really easy to understand. I thought it was, really exciting. And eventually, this is the lab I went to for my postdoc. But along the way, I went down another path and studied the auditory system to study pitch perception in non-human primates for my PhD. So towards the end of my PhD, I was thinking, more about memory and how sequences of sounds might be processed. And I shifted to study the rat hippocampus because it was an exciting area where there have been a number of papers looking at this replay phenomena, and in much more detail with recording methods that were much more sophisticated, allowing you to record from many neurons simultaneously. So I started looking at all these replay studies. I joined the lab, and fortunately, when I joined the lab, I think within a year or two of me joining, a paper came out on targeted memory reactivation, which used auditory cues when someone is performing a task. And when you repeat these auditory cues when they're sleeping, the performance in the task after they wake up improves. And this combined my interest of the auditory system from my PhD with replay and sleep, which is what I was working on for my postdoc. And so that was the perfect stimulus for me to try to go after these sorts of experiments in rodents. And we wanted to do this because in humans, we had some hypothesis of the mechanism, but we wanted to connect the dots. So replay is thought to be this phenomenon of repeated neural activity during sleep that matches the neural activity you see during an earlier behavior. It's thought that this repeating of neural activity is important for learning and memory consolidation. So the thought is, if targeted memory reactivation in humans improves a memory by reinforcing it during sleep, we should see some manifestation of that in replay. So more replay of the memory being reactivated, which would then lead to better memory consolidation.
And the idea with the targeted memory replay is that the sounds actually pair somehow to the learned information. And so representing those sounds during sleep actually leads to somehow triggers or biases the brain to reactivate those particular memories that were associated. Is that right?
That's right. I mean, the first paper with targeted memory reactivation was with olfactory cues. And there seemed to be the possibility that that was just activating the hippocampus directly and causing more activity and then strengthening the memory. But with the auditory cues, there's not so much a direct pathway. So it allowed us to explore the hypothesis that somehow the activity in cortex might bias the hippocampus to replay particular memories. So it's not simply when you activate the hippocampus in some way, it causes stronger memories. What we think is happening is that by activating certain regions of cortex, you prime the hippocampus. The hippocampus is potentially choosing which memories to replay, and it gets primed by activity in cortex when you play a sound to replay a specific memory. And now I can get more into this later, but now we're exploring why is this the case in the first place. So is targeted memory reactivation essentially piggybacking on top of existing mechanisms that help the hippocampus choose what to replay? So if I gave you a behavioral task and then I tell you, actually, just the second part of the task, you'll get paid $1 million tomorrow if you do really well in that. In the first half, don't worry about it. I'm not going to ask you again. Then I actually lie to you and I test you on both. You'll do much better on the part that you associate with a higher reward. And since I only told you that after learning, the assumption is that somehow the brain prioritizes memories that it deems to be important for the future. How that happens, we don't know, but we suspect, or at least hypothesize, that there's a similar mechanism. Targeted memory reactivation plays a sound and tells the hippocampus, this is important to replay. And normally that's just going to happen sort of organically through spontaneous activity, that parts of the brain associated with important memories are going to be more active and tell the hippocampus, hey, I need a memory replay back to me. And then the memory gets strengthened.
So what you're saying is that there's a hierarchy of memories in terms of which ones are going to get reactivated, and the reactivation influences how they get consolidated, and that the targeted memory reactivation somehow hacks the hierarchy and biases the system towards replaying the memory that you've chosen, that you've targeted in this way. Yeah. So I feel like there's a lot more we could get into there, and we will, but let's back up first and talk about this, the experiment that you did with the rodents and the targeted memory reactivation. And maybe it's worth premising it by saying that I believe your study was the first one to attempt this in a non-human, right? So there had been a number of studies at that time in humans showing that playing these sounds or presenting odors that had been paired with learned information during wake would lead to reinstatement of the memories in sleep and facilitate subsequent performance on remembering that information. But no one had tried it in animals, and consequently, no one had really looked at the neural basis in a careful way. I mean, there was a bit of fMRI data, a bit of EEG data, but nothing looking at the population or a single neuron level at all. And I think that's really what the animal work can add. So maybe you can talk a bit about that.
Sure. So there are a lot of advantages studying humans. Okay. So first, you can train them on much more complicated tasks than you can for a rat. So for targeted memory reactivation, you usually have, like the paper that came before my study had 50 different pairings between sounds and objects and locations. Okay, so it's an association task. And 25 were presented during sleep, those sounds, and the other 25 were not. There's no way we could ever do that with a rat. So for a rat, we had to simplify it to the most basic form, which is for one sound, it runs to the right side of a linear track. It starts at the center. And then for the other sound, it runs to the left side of the track. And to avoid just the animal turning right, turning left, it can initiate the task from the center, but from one of two locations. So it's facing the opposite direction. Instead of turning right, it has to go to the left. So it's basically the right side of the track is associated with the sound rather than turning right or turning left. And that might seem like a very minor thing, but for rats, they can have stereotype behaviors or sort of other things that complicate it. So essentially you have the animal running right, running left along a track. And then when it goes to sleep, we know in the hippocampus that neurons are tuned to the location of space that the animal is in. Each neuron likes a different location. So, you could imagine that each cell, if you paired that cell with a musical note on the keyboard, then when the animal runs right, you'd hear something going up a musical scale. And when it runs to the left, you hear it going down the musical scale. And when the animal goes to sleep, you hear do 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 do. So you can hear the animal is running to the right or running to the left just from the sequence of neurons that are active. And it's the same sequence when the animal is running on the task as when it goes to sleep. And so as I said, with humans, you have more complex behavior. But with rodents, because we use electrophysiology, we can implant many microwires that allow us to record from 50, 100 neurons simultaneously. And that gives you a very good resolution of what's happening. That when you see a sequence of neurons firing in a rodent brain, and you're recording from the same neurons over many hours, that gives you a resolution to know, yes, this pattern is very specific and it can't happen by chance, right? The chance that 100 neurons fire in a perfect sequence is extremely, extremely small. So it gives us statistical certainty. It gives us a resolution of seeing these patterns when they're occurring without any delay. And as a result, we can then measure reactivation patterns of the animal running to the right, running to the left, and then see how they're modified. So when the animal was sleeping, we could play one of the sounds. So let's say we play the sound associated with the animal running to the right during the behavioral task. And then we can measure, is there more replay of the animal running to the right or more replay running to the left? And if we didn't play a sound, there should be roughly equal amounts of replay to the right and to the left. But we found that when we played the sound to tell the animal go right, there was a bias towards running to the right over running to the left in these reactivations. So we don't know if the animal is dreaming, but it is reactivating a prior experience, and it's reactivating experience associated with sound much more than other possible experiences. And the same thing happened if we play the sound saying go to the left side of this track, then we see more replay to the left than to the right.
So can I simplify it? Tell me if this is accurate by saying you paired sounds with right and left hand sides of the track during wake while the rat was running around exploring, and then you replayed either the right or the left sound in sleep. And then because you're recording from place cells during the sleep, and because the place cells are associated with those different areas of the track, so you know which place cells are associated with the right and which are associated with the left, you could look at which sequence of place cells fired after you played this sound, and you could tell that if you played the right hand side, then the play cells associated with the right-hand side would fire. And if you played the left-hand side, then the left-hand side ones would fire.
That's right.
And can I just ask, so in terms of those play cells firing, was it sort of a series of play cells associated with each side, or was it just, I mean, were they playing as if it was running along that track to the right or to the left in order, or was it just kind of a conglomerate of play cells associated with right or left?
Yeah, so the replay phenomena you see, it's ordered. So, if you, let's say you had 5 cells, A, B, C, D, E, we're going to call those cells A, B, C, D, E. Then during a reactivation event or a replay event, these cells will fire A, B, C, D, E. However, the brain is noisy, so it doesn't always fire A, B, C, D, E. Sometimes it will fire, slightly out of order, or one cell won't be active during the reactivation event. And so it involves a lot of statistics to figure out, can these reactivation events happen by chance or not? And so as you have more and more cells that you're recording from on the track, you can say with greater certainty, if it fires in a sequence, that this just can't happen by chance. And just to give an example, if I only had 4 neurons I recorded from, and they fired in a perfect sequence, it could still happen by chance a small percentage of the time, like 5% of the time. And so you really need a lot of cells in order to say, yes, this can happen by chance. But it's challenging because the brain is noisy. You have to make sense of, if something is firing out of sequence, is it noise or is it serving another purpose? So for instance, there have been studies suggesting that sequences the animal hasn't actually experienced can occur to represent a shortcut between 2 points, or maybe building a model of the environment. So there could be aspects of replay we don't fully understand. That's not simply repeating an experience. And we have to be careful with our interpretation that just because the sequence is not repeated exactly the way we expect, that's not necessarily noise. But it also can be. And so we have to run the right experiment to tell the difference.
Yeah. And doing this in rodents really added, that was, exciting to me anyway at the time, was the fact that previously in the human work, people had been playing these sounds and seeing a behavioral benefit after sleep. And yes, there was some EEG evidence and some fMRI evidence that replay might be happening after the sounds, but no one had been able to actually really index that in any kind of convincing way. And this study of yours was the first one that showed really when you play these sounds, we can see at the neural level that this replay is actually happening, and it is associated with the sounds. So for me, that was super important. It sort of took it from the level of hand waving, we think that the behavioral benefit we see after sleep might be associated with some kind of reactivation in sleep, to say, no, we can actually measure it. And you measured it, which was really cool.
I mean, in neuroscience, a field usually starts off with phenomenology. So, for 15 years, even more 20 years before I started the project, people had found evidence of replay in different areas of the brain, mainly the hippocampus. But it was very difficult to say this replay is very suggestive of there's a sequence occurring offline. It's a memory trace. Therefore, it's probably related to memory consolidation, because sleep is related to memory consolidation from human work. But connecting the dots is very difficult to show causally that this is important. And before I joined the lab, there were 2 very nice studies that disrupted hippocampal activity and demonstrated that if you disrupt hippocampal activity during these replay events, so there's sort of a marker of when these events are occurring. There are these high frequency oscillations in the hippocampus called sharp wave ripples. So if you disrupt the hippocampus during these sharp wave ripples, presumably that's disrupting these replay events, we see a deficit in learning. So they're really beautiful and difficult studies that happened. But there's always you could argue there's always a concern that if you disrupt brain activity, it's hard to know exactly what are all the consequences. So I wouldn't say this was definitively showing a causal relationship, because we didn't record the behavior afterwards. That was technically difficult to do. But we're trying to show in a positive way that if you can actually have more replay events, and at least in humans, doing a similar task that's associated with stronger memories.
Yeah, super, super cool. I believe you also showed an additional interesting thing here, which was that it wasn't just one replay that was happening after the TMR. It sort of continued to occur until there was another cue biasing the replay towards the other side. Is that right?
Yeah, that's correct. So I think an assumption or a hypothesis we had early on is that if you play a sound, you will just affect the next replay that occurs. But what we found is that the sound you play creates a state. You can think of it as a state in the brain that as long as that state is there's a constant bias towards playing that sound. It doesn't seem to go away till another sound was played. We only tested it up to 10 seconds, but 10 seconds is quite a long time for brain activity. That if I play a sound and then I stop the sound, neural activity in auditory cortex will, generally speaking, in the vast majority of neurons, goes away fairly quickly. It's not going to last 10 seconds. And so something else is happening in the brain that somehow saves this information and says, okay, keep on playing replay events associated with that sound. It does match up with human literature as well. So we don't know how long it goes for, because 10 seconds was the maximum amount of time between stimuli that we used. Presumably it would go away at some point, but we don't know how long.
Yeah. And so this takes us back to what you started to touch on earlier, which is this idea, you just used this word biasing. So it's not so much that the targeted memory reactivation is triggering a replay as that you would argue it's kind of hacking the system and biasing the brain to replay the memory that you've cued. Can you talk a bit more about how that works?
Of course. So this is extremely speculative without evidence. But we do know that there are up-down states in the brain during sleep. So that just means there are periods where neurons are extremely excitable and then periods where they're very sluggish or silent. Okay. And that's going on during non-REM sleep, where these replay events are occurring during these shortwave ripples. And so during an upstate, we have neurons across all of cortex suddenly very excitable. One possibility is that they're not all equally excitable. It's just a state where they can dynamically, let's say, make decisions of which part of the brain is going to be more excitable than other parts of the brain. So it's like you're taking your foot off the brake, and now with the accelerator, your car can go fast to the right or fast to the left. Okay, so it allows fast dynamic changes in the brain. Now, if there is one part of the brain that is more excitable for whatever reason, let's just say for now, we don't know what the cause is, it could potentially outcompete other parts of the brain and then tell the hippocampus, replay back to me. When we play a sound, we think we are causing artificially one of the brain areas to outcompete other brain areas. But normally what would happen is that one area would be more excitable because that memory was more salient, and it tells the hippocampus, okay, replay back to this neural circuit. And you could ask yourself, well, if there is some form of the memory already there, why does it need the hippocampus? And one possibility is that aspects of the memory, the different associations, are stored in cortex very quickly. But the sequence of events is something that the cortex has trouble doing on its own. The hippocampus is very good at that. We see sequences being activated, reactivated all the time. And so the hippocampus might be thought of as a teacher saying, you have all the bits of the memory, this is how you link them together. And so it needs to be trained. And a memory in cortex will say, okay, I need training. The hippocampus then trains that memory. And then this keeps on going during sleep.
So it's a dialogue between the cortex and the hippocampus. And so there are several things in what you said that I have to know more about. So first, you said that the cortex says to the hippocampus, I need training on this. So it sounds like it's initiated, or at least you're thinking the dialogue is maybe initiated at the cortical level. How would that work? How would it know that it needs training? What kind of thing might it need training on? You mentioned the idea that maybe the cortex is trying to assemble together various components of something, and the hippocampus is saying, you have all of these components. How does that dialogue work?
Yeah. So if we come back to the analogy of the brakes and the accelerator in the car, this is something that you see in each neuron, that there's excitation and inhibition. Excitation is the accelerator, inhibition is the brake.
Are these the slow oscillations, by the way, or these are the up states and the down states in sleep?
No. I just mean in terms of the inputs to the neurons. You can scale up synapses so that the neuron is more excitable, or you can scale up inhibitory synapses so the neuron is more sluggish. And without getting into the details, if you think of the car analogy, one way of encoding information is to press on the brake and the accelerator at the same time. So if you do that, the car moves at the same speed. So it has the same net excitation. But as soon as you remove your foot from the brake, the car goes really, really fast. So there are ideas that you can have neurons respond in a similar fashion, but what you've done is you've scaled up excitation and inhibition so that they're sort of holding a memory that when the time comes, they can, when you remove the inhibition, they can be way more excited than other neurons. If that's the case during up and down states, it's possible that neurons that seemed really similar during behavior, one of those neurons is scaled up in its excitation and inhibition. As soon as you remove the inhibition, it becomes way more excitable. So there might be a way of storing information in some neurons based on how relevant they are or important they are in the future. So if you pair a sound with a reward or you shock the animal so that it says, oh, this sound is really salient, the neurons should scale up their excitation and inhibition according to this. And so the way the neuron responds when you play the sound will look similar, but as soon as you remove the inhibition, now the neuron is way more excited than all the other neurons responding to other sounds. If that's the case, then during an upstate, this neuron that is important is more likely to be active, or it'll be more active than other neurons and send a signal to the hippocampus. I need you to teach me how am I involved with all the other neurons in the brain for this memory.
Okay, so the idea is that the targeted memory reactivation scales up the excitation. So this is like really stepping on the accelerator in the cortex. And then the neuron that is kind of hyper active and going really fast is sending a signal to the hippocampus going, I need to know what I'm connected with. And so it's saying basically, teach me or give me information. And that triggers a reactivation or replay in the hippocampus. Is that right?
That's right. So the other analogy you can use is each neuron is like a member of an orchestra. So it might know its part, but it doesn't know how it fits in with all the other instruments, right? So if each instrument played correctly but not together, it would sound terrible. And the conductor is there to make sure that in a coordinated fashion, you play together with the other instruments when it's time, or you play in a sequence that one instrument happens and then another instrument. So the hippocampus is the conductor, and the instruments, when they say, oh, I'm really important in this musical piece, I need to play loudly and get the conductor's attention. And then the conductor trains all the musicians that have done this together, so that you get a nice sounding piece.
Yeah, it makes perfect sense. But then what I'm trying to understand is this interaction happens and the hippocampus responds by triggering. I mean, what is the response that happens in the hippocampus? It's a replay, but does it replay not only the things associated with that neuron, well, directly associated, but all of the more indirectly associated things with that neuron as well, like all of the context, all of the other players in the orchestra? The hippocampus will replay at the same time. Is that
right? So one thing the hippocampus is very good at is something called pattern completion. That if you give it, let's say you see a picture, a fuzzy picture, but there are a couple of cues and you say, oh, okay, I recognize that. That's where I went on holiday, 5 years ago. The hippocampus can take inputs from some parts of cortex, and then with feedback, it can activate presumably all the other parts of cortex involved in that memory. So it's completing the memory and then filling in the details. And it does this because cortex is connected to the hippocampus and hippocampus is connected back to similar regions of cortex. And so that's maybe something necessary for targeted memory reactivation to work, that you give one cue out of all the other cues associated with the task. And that's enough to drive hippocampus to reactivate all the necessary players of that memory. And once that happens, then you have sort of a state in the brain where that memory is being enhanced in both hippocampus and cortex, and a sequence can then be reinforced.
Mm-hmm. And so the hippocampus reinstates the whole orchestra in the hippocampus, and then it connects to the cortex and reinstates the whole orchestra in the cortex, and then the whole orchestra is kind of playing, and then presumably those two structures continue to reverberate back and forth like this, and that will strengthen the memory for how all of these instruments fit together and the temporal sequence in which they do that as well.
That's right. So one thing in the hippocampus that's perhaps really critical is that if I take a rat and it's running around, in the room, each neuron I record from will have a specific location in space that it likes. If I take the animal to a new room, there's a phenomena called remapping, where now each neuron will like a different location, or perhaps be completely silent, or neurons that were silent in the first room will suddenly be active. You can think of it as you're just scrambling the preference of each neuron when you do this. But the advantage is, just by looking at which neurons are active, if I was sort of peering down at the brain, I could tell which room the animal is in. So it allows me to say this pattern of activity, it's like listening to the musicians in an orchestra. If I only hear strings, I know I'm listening to this particular orchestra, and a brass band will be completely different. It's different players, and I'm over here. And this allows, you can think of it as like a railroad track. You're selecting, do I go left or do I go right? The hippocampus does that disambiguation. So it does a very good job at separating out context, even if in cortex, very similar neurons are being involved. So there's more pattern separation going on in hippocampus, which you need if you give one cue and you want to say, okay, this is the state. Even though, both environments had very similar cues, I know I'm in room B and not room A. And now that so you can think of it as the conductor, another analogy. You can think of it as the conductor. Conducting, there are two pieces that have similar overlapping melodies, but the conductor knows which piece is being played and directs the instruments accordingly.
Yeah, it's very intricate. I mean, it's a kind of a hard concept to get across, but I think this is clear. So this is the theory, right? I mean, this is how you're thinking of it, and some other people are thinking of it. I think I agree with you by and large. What is the evidence supporting this?
We know pattern separation, pattern completion, remapping, those phenomena are well studied. The up-down states are well studied. The fact that there is a bias in cortex that leads to the hippocampal state, there is much less evidence. The best evidence is probably coming from Gideon Rothschild and Lauren Frank, where they showed auditory cortical activity could predict what would happen in the hippocampus during replay. And then vice versa, that replay would then tell you what is happening in cortex. So it shows that there is some sort of intricate interaction between the two. And it's not simply going in one direction, it's going back and forth. There's a dialogue. But the nature of that dialogue is something that is actively being investigated. That's something that we don't know. So is the cortex really telling the hippocampus, you need to replay this next? Or is it something more complicated? And secondly, when the hippocampus sends information to cortex, how is that actually integrated? We assume that a teaching signal will cause the student, the cortex, to practice the same sequence. But beyond that, we don't really know, is it going to a particular part of cortex, and what happens after the training signal comes? There are a lot of things to be filled in.
Mm-hmm.
And there's also a third part that's really important that I think generally has been overlooked, which is when you've replayed something enough that a memory is effectively stored or close enough to being stored, you would want to perhaps bias your replay towards other memories that are not as important but still important. So how does the brain decide, I've replayed this enough, or I need to replay this more? Like if you wake someone up in the middle of sleep, does the brain have some mechanism to say, actually, you didn't replay this enough, you need to replay this more? So there has to be some sort of feedback, and that we really don't understand. So like, is the brain measuring how well the memory has been stored, or is it simply just replay something 200 times and then that's it, regardless if it was, good quality replay or not?
Yeah, so there's a question mark around essentially when it decides to stop replaying. It seems very unlikely that it would be a fixed number. It seems much more likely that there would be some kind of a quality measure and threshold. But of course, we just don't know what the mechanism would be for that.
Sure. And I mean, a fixed number would be simpler from a mechanistic point of view. But yes, it wouldn't be a good mechanism. But you never know what the brain picks. I mean, there's always, all these things have to develop, through evolution and through brain development. And, something is not feasible, then sometimes there's a suboptimal way that's chosen.
Yes, but a fixed number doesn't sound very biological.
No, but we do know that it's time limited. So the experiment I said, where you tell someone after the task, this is going to be really important for you to remember. If you sleep deprive them long enough, and then you allow them to sleep as much as they want, the bias related to the reward is lost. So you have to sleep within, I think, 48 hours, 24 to 48 hours in order to benefit from that. And that suggests that the memory might not be gone, but the bias is time limited. And what we don't know is presumably if you do sleep, then the bias also disappears because you've stored the memory. So that allows you to store other memories.
Yeah, I think that question of how long the bias lasts is really interesting. So for instance, there are a number of studies. There's one from Scott Kearney and Bernhard Starosina where they applied TMR during a nap, and they saw absolutely no impact of the TMR in testing after the nap. But then the participants went home, slept for a normal night, and they tested them again the next day. And then they saw a benefit from the TMR at that stage. So I remember when I first saw that, I didn't really believe it. I just thought, something really dodgy going on in these data. But now we've seen that kind of pattern again and again, where another example is sometimes the TMR, the targeted memory reactivation, is applied during slow wave sleep, say, but we actually find the benefits of it are predicted by the time spent in REM sleep. And so the interpretation is this bias, as you call it, to reinstate that particular memory is continuing across time, and it's reinstating in REM sleep. And in the case of the Kearney and Starosina paper, it's reinstating in that subsequent night of sleep. So this is a really interesting phenomenon, I think.
Yeah, and I agree. I mean, the role of REM sleep is even more mysterious in many ways, and it is hard to study in rodents. So humans, I think about 20% of the night is spent in REM sleep. In rodents, it's, 5 to 10%. And also the way sleep is detected in rodents, it's much more sophisticated in humans in terms of different stages. And in rodents, we can see that they're in something like non-REM sleep, but we do not distinguish between stage 2, stage 3, stage 4. And so that does make the comparison between humans and rats a little more difficult, but we can distinguish between REM and non-REM very easily in both in humans and rats. And so most of the work has been in non-REM. This is the sharp wave ripple replay. And there's been one published study in REM sleep in rodents, Louis and Wilson. And there they see evidence of replay. It is one important thing to note is that REM replay in rodents, the speed of the sequence happens similar to the speed of the sequence during behavior. But in non-REM sleep, it happens 10 to 20 times faster. The reason that is really important is that for two neurons to strengthen their connection, there's a certain time window that they both have to be active. So this is something called spike time dependent plasticity. And it has to be really close in time. So if one neuron fires, another one fires 1 second later, nothing's going to happen. So when you temporally compress a reactivation event or a replay event, it forces neurons to fire very close in time and then strengthen their connection. Well, that's a mechanism that would work very well with memory consolidation. If that's the case, we don't know what REM sleep is doing, but because it's happening on a much slower time scale. But it could serve other functions that are important for memory consolidation beyond just strengthening synapses.
So can I just check I'm understanding here? So what you're saying is the replay that happens in non-REM sleep is quite fast. So it's very much sped up compared to the time that those same events would have taken during wake. So if a rodent is running down a track, then that's sped up. Is it something like 21 times faster in that range?
10 to 20 times. So basically, one of these reactivation events is about 100, 200 milliseconds. And so that represents anywhere from 1 to 4 seconds of animal behavior. And if the animal is running over a longer portion, so let's say it takes 20 seconds for the animal to go from one side of the room to the other, you'll see that there are multiple replay events that happen sort of in chunks. Boom, 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 boom. So there's only so much information you can compress into one, and then you just get a chain of these events. And at the sort of the unit of memory expression in the brain during non-REM sleep.
Yeah, so the non-REM reactivations are very fast, and that's good for spike timing dependent plasticity. And so it would allow kind of binding together of those memories with this plasticity. But the REM reactivation that has been identified, and there's only this one paper from William Wilson, although I know this has now been replicated by...
In Matt Jones's lab.
Yeah, Matt Jones's lab. So not published yet, but watch this space, has been replicated. So that reactivation in REM was slower. It was only sped up something like 6 or 7 times compared to wake, I think. And so what you're saying is it's less clear from the spike time dependent plasticity perspective what that would be doing, but it's probably still doing something. Do you have ideas about what it's doing?
So there are a number of different ideas, and since there is, at least in rodents, little evidence, it's very easy to speculate if you're not tied down to specific evidence. So first of all, I should say that when you block REM sleep, there have been studies that show that there is no effect, and other studies that show that there is an effect on memory consolidation. And if you look, the second important thing is if you look in the animal kingdom, there's some species that have almost no REM sleep. Dolphins, for instance, have minutes per night, and it's very short bouts because they can't breathe during REM sleep, so they would drown if they were having REM sleep for 20 minutes at one go. And if you look at the animals that have a lot of REM sleep, they're animals like the opossum or the ferret, not necessarily the most brilliant animals with the best memory. So the question is, if it serves a function, why do some species, like dolphins, are pretty smart? They have very little. What is the benefit that they're losing that the opossum has gained? With behaviorally, we don't. I mean, maybe opossums are brilliant in some tasks that we haven't tested. So from that point of view, it makes it very mysterious what REM sleep could be doing. It should have some advantage that you still can manage without it. Alternatively, it could be that dolphins or people that have lesions to the brain and don't show REM sleep, they find some alternative to perform the same function, and we just don't know what that is. So some ideas. So REM sleep is associated with dreaming, even though you do dream during non-REM sleep as well. But there are lots of stories of people that solve puzzles in their brain when they're sleeping. So REM sleep could allow exploration of, you have lots of information from the world. You build an internal model gradually saying, I think this is how this works. I have all these little snapshots of evidence I'm putting together to have sort of a larger idea of how things work. REM sleep could be instrumental in building these models. Now, you can function without a model. And so it could be that maybe dolphins are very good at what we call model-free learning. And so it's not critical for them. But for us, it's beneficial. Second thing is that non-REM sleep is important, we said, for taking a memory in hippocampus and storing it in cortex. But there's a second component that's important, which is being able to recall the memory without the hippocampus anymore. The hippocampus is very good if you give it a little bit of information. It does this pattern completion and tells you where the rest of the memory is. But if you lesion the hippocampus, you can still access very old memories, which means you can piece all these things together without this brain region. It's possible that what REM sleep does is connects together brain areas so that you don't need the hippocampus anymore to recall it. And that could be one explanation why we have all these bizarre dreams that are related to perhaps your emotional state from the day and recent memories, but also tend to use much older memories to represent your dreams. The bad news there is if you're representing very old aspects of your memory, we're going to have a hard time finding out what these are in the rodent. Because when you do these replay studies, you typically put the animal on a track. And if the animal replays running back and forth on that track, you're golden. You have a template you can match and say, I know what the animal is replaying. If it's replaying something else that's completely different, you can't interpret what you're seeing. So that makes the experiment more challenging. We'll put it that way.
Certainly in rodents, it does. Yeah, we may come up with ways of decoding that kind of information in humans. So basically what you're suggesting is that REM replay might be more involved in integrating current learning into past experiences, but we'll have a lot of trouble identifying that it's the past experiences that are being replayed. It's very interesting. I mean, do you know of any, are you working on this yourself at the moment?
So we are starting to do some REM sleep studies. I think the key thing is being able to figure out what these reactivations mean. And since we want to not use a sort of template method where we say, okay, this matches very well to this. And so we're going to experiment using machine learning approaches to see if we can decode these patterns more effectively. Now, I have to say that one very difficult thing with this field is that we don't have what's called a ground truth. So if we see a pattern, we don't know that this pattern, regardless of what technique we're using, we don't know that it actually is something. So when the animal is running around, we can track where the animal is physically in space. We can look at the neural activity and then show that our algorithm can predict where the animal is at any point in time with a certain degree of accuracy. That is a ground truth. When it's dreaming, we can say it looks similar to this, but we don't know with 100% certainty that it is actually dreaming about this event.
Yeah, so sure. So this is the tricky decoding question, but presumably, and if you're working on it, you have some workaround for this. So you have some way in which you're going to try and identify what the rodent is reactivating, and then somehow test whether it's doing this integration function.
Right. Yeah. So if you have a match, so if you have a template, you can say activity matches the template with high certainty based on the statistical chance that it could happen randomly. But as soon as you deviate from this template, that's where it gets trickier. There are potential workarounds that are not perfect, but essentially you have to track what the animal is doing. If you can record long term, let's say over 1 or 2 months, and figure out everything the animal has done, then maybe it's possible to then decode.
Oh, okay. It sounds very complex. So you have some really fascinating sounding papers that I haven't yet read. But so, for instance, you have one about how the hippocampus maps the future. Could you talk a little bit about that?
So this is critique on the field. So there's a debate in the field whether activity in the hippocampus, if these replay events, if they can occur prior to the animal actually experiencing the track. So one side of the field says replay is a byproduct of experience. You experience something, the hippocampus stores these experiences by the sequence of neural activity that represents the behavior. And then, only then, when you sleep, these patterns reactivate for the purpose of memory consolidation. A second part of the field thinks that these sequences are already built into the hippocampus and they occur prior to the experience, and then perhaps they form some sort of scaffolding structure that memories can then be built on. And in this paper, Hugo Spears, we made the point that when you look at replay activity, you can actually figure out the topology of the environment. Topology or topography, I always get that confused. But essentially, you can imagine if the animal was running around a circular track and repeating this, you would get a pattern of cells A, B, C, D, E, A, B, C, D, E, and it would repeat in a circle. There is no start or end. That the E cell would connect back to the A. If the animal is on a linear track, you would get a pattern A, B, C, D, E, or E, D, C, B, A. But the E and the A never get connected together because the animal would be teleporting from one side of the linear track to the other. So based on that, you should be able to look at neural activity and say, is this a circular environment or is it a linear track, for instance? And there are other possibilities of other environments. So this has been shown already that just from the sequences of neural activity, you can guess which locations are neighboring each other and which ones are far apart. And we felt that was enough to say you cannot know the topology of an environment before you're actually in it. So you can't have sequences that exist prior to the experience. It might be possible for 1 or 2 cells, but not entire sequences, because if that was the case, the animal would know what environment it's going to be in, which it doesn't. I mean, if rats can predict the future, then we should be studying them for investing in the stock market. But it's we can't. And the second argument is that, as I said before earlier in the interview, if you put the animal in another room, cells that were nearby each other are the place fields now remap, and they will be very far apart. Well, if the animal when it's sleeping doesn't know what environment it's going to be in, it can't predict which cells should be next to each other and which ones should be far away. So we think that it's likely that the methods used to detect replay are perhaps making mistakes statistically, and things that are happening they're saying it can't happen by chance. It can happen by chance. and we need more rigorous techniques. And it's not to say that it's a easy thing to do. It is really difficult. That you tend to take the data and shuffle things around to show that it can't happen by chance. But each time you do that, you're making certain assumptions, and you can be more rigorous, but sometimes it's at the cost of, if you're too rigorous, then you actually are missing a lot of real events as well. So there isn't a perfect method out there, and so labs are debating about how rigorous they should be or not.
Yeah, I mean, it's the classic kind of trade-off in science, isn't it, between type 1 and type 2 errors?
Yeah,
so,
exactly. And
yeah.
so the hope is maybe with machine learning or newer approaches, we can solve that. But as I said before, we don't have a ground truth. So if I show you what I think is a replay event, you can say it matches this sequence really well, but you'll never know that it is actually replaying that, because we don't know what the animal is actually thinking.
Yeah, that's right.
Other brain areas, right? We don't know what its perception is, right? It's a mental experience.
Yeah, you only have the measure that you have, and we have to extrapolate from that. Okay. So you also have a paper about how hippocampal dependent memories can be enhanced, deleted, or integrated. Can you talk a little bit about the ideas in there?
Yeah, so that was a review where we were looking at sort of all the latest approaches to manipulating neural activity. And I think over the last 10 years has really been an amazing progress in the field in that respect. So, there are approaches using optogenetics, citing particular neurons with light, so which you have very good temporal control over their activity. And this is a genetic approach, transgenic approach. So you can cause only neurons associated with a particular memory to be sensitive to the light. And it's a very cool technique developed in the Tonegawa lab at MIT, where, an example, you take the animal, you shock it in one environment, and normally if you put it in a second environment, it wouldn't freeze. It only freezes if you put it back in the first environment, because it remembers, oh, I got shocked here. That's not good. And freezing is a natural behavior for the animal when it's scared and it can't escape. But what they do in the first environment is they tag the neurons. I won't go into the details of how, but they tag the neurons in a way so that when they shine the light in the future, the same neurons that are active during the freezing behavior or the shock become active. Now, when they put the animal in the second environment but turn on the light, now those same neurons are active. And even though the animal is physically in context 2, it freezes like it was in context 1. And they've done a lot of different variations to this experiment to explore these what called engram neurons. But they've shown that you can tag neurons associated with a memory and then turn them off or silence them and then this is a way of studying how memories get encoded in the brain. So you have sort of that approach. You have targeted memory reactivation, which in humans is easier to do than sticking viruses for transgenic expression and attaching a laser to their skull. It's a much more, non-invasive approach. So there's, an amazing amount of work has been done over the last, I guess, more than a decade, looking at all the aspects of this. And then there's sort of other ways to manipulate memories, but essentially turning the memory on, turning the memory off, or changing the meaning of the memory. So another example, they tagged a memory that was negative. This is also work in the Tonegawa lab. They tagged a memory that was negative, and then they reactivated the memory with something positive and removed some of the negative valence. And in a second group of animals, it did the opposite. They took something positive, they tagged those neurons, and then they shocked the animal while shining the light to activate those neurons, and they removed the positive valence. So that suggests that not only can you cause a memory to reactivate somewhere new, but you can actually change the meaning of the memory by reactivating and associating with something new.
It's so amazing, isn't it? So it's kind of brainwashing in terms of associations. But this doesn't involve sleep at all. So I'm going to pull you back to sleep. I guess we're starting to get towards the end now. So let me ask you, what should I have asked you? What are you working on now that you want to talk about?
So I think the replay field has really grown tremendously from, roughly when I joined the field, there was this very rapid growth, and now many labs are studying it. But still largely the majority of studies have been looking at the phenomena of replay, like basically what happens if you have a longer track, or where replay is occurring, what brain areas can it happen when the animal is sleeping or awake, does it happen in the forward or reverse direction. So it's all of these things. We're trying to get a better picture of how replay manifests in the brain. And the quick summary is that you see it everywhere. It seems to be a very robust phenomena, and it's much easier to see if you have the right recording tools, which allow you to record many neurons simultaneously. So for instance, the Foster lab at Berkeley is probably the leader in the field, where they have, I think, 40 tetrarods simultaneously. So that's 160 microwires in the brain, and they can record from 200 neurons simultaneously. And that gives you a level of precision that allows you to see a much more than we could 20 years ago when it was 6 tetrarods at a time, right? So that's one reason the field has exploded. The second is that it's not just sleep, which is harder to record, but when the animal is awake and doing a task. So all these phenomena of replay have now broadened our expectation of what replay might be able to do. So it's not just for memory consolidation. It could be for planning. So the animal's deciding what it wants to do next during the task. It can reactivate. Do I go right? Do I go left? It could be also, another hot topic now is it reinforcing something that it has done well, or is it building a model of the environment, which is reinforcing not just the good, but everything. So it now has a more complete map of, if I want to take a new path, where do I go? And we don't know. I think a big question is we don't know the difference between these reactivations that occur when the animal is awake versus sleeping. That's one big question. And the second is, how is the replay being used? If it's for memory consolidation, how does it actually decide what to replay, and how does it actually change what is going on in neural circuits? How is it modifying neural circuits so that they're storing the memory in a different way? Those are hard questions, so they take a long time. But that's really the next step. So and once we know that will allow us to look at these mechanisms more deeply. So for instance, in reinforcement learning, we want to understand how the brain figures out, oh, I went this path, I took this path. This is actually the better way of going. That's a hard problem. But for people doing, machine learning, reinforcement learning algorithms, they would like to model how the brain does it so well. And so there's a lot of reason for us to explore this.
So why should we care? Why do we want to understand this system?
I think what makes the human brain so interesting is its ability to learn, not just simple associations, but sort of complex tasks. And, it's not just saying, I got shocked, I was next to the green light over here, green is bad. It has to build much more complex models of the environment where in this situation, this is bad, but only if this happens. How that all comes together is if we come back to the orchestra metaphor, right? It's going from playing a very simple instrument to playing in an orchestra. How do you coordinate all those things together to learn cohesively? Like we want to play this piece. And there's so many moving parts that it is difficult to simplify an experiment to the point where one result will give you an answer. So it's also a bit of an orchestra between labs to come to a consensus.
So in some sense, it's, just the search for knowledge, isn't it? Why we're interested in, understanding how the brain learns. I mean, I personally just find it fascinating. And for me, I know that what drew me to the field is this idea that this is happening offline while we're not paying any attention, these replays and consolidation. But I guess backing away from it a little bit and thinking about society, how's this information going to be used? And could it even be used in a negative way? Should we be worrying about this? Should we be worrying about brainwashing? I get asked sometimes with the targeted memory reactivation studies, is it really good if we can alter people's memories while they sleep?
Yeah, I don't think we have to worry about brainwashing because no one is forcing you to listen to sounds when you're sleeping, right? And the beneficial effect of that, it's not like you don't, it just causes a slight enhancement in what you're able to remember. right? So it's not like the optogenetic experiments I told you where you are reactivating a specific memory. The sound has to go through multiple pathways in your brain, and the effect is significant, but it's not overriding other things. And I think we should be much more concerned with what we're doing in our daily lives. I'll give you an example. If TMR is basically biasing memories towards the sound, but normally it's what is important or salient that drives memories. We know that we're on our phones looking at Facebook or Twitter all the time. And it's quite possible that when your brain has to decide, what do I replay from today? It is biased towards novelty. And what is novel is all the junk information you're digesting from your phone. So it has two sides. First, we're putting lots of junk information. So it would be like eating junk food, right? So junk food has all the salt and the sugar and the fat that your body craves, because back in the caveman days, those things were hard to get. Now we can make junk food that just has that, but without the nutrients. And the information we digest from our daily lives does not have any nutritional value, but it has lots of novelty, which we seek. So we're addicted to the novelty. It's probably overriding these pathways in our brain to say, look, you really need to remember your Facebook feed rather than important things from the day. And the second part of it is that we're on our phone so much, we're probably affecting our sleep patterns. People are looking at their phone in bed, even the light from the screen can affect sleep. And we're sleeping less and less as a society. And it's not just that memory consolidation is affected, right? That we're remembering unimportant information that was novel, and we have less time to do it. So we're pinching the system from both ends. On top of that, sleep has been associated with Alzheimer's disease and many mental disorders, that when you have less sleep, it leads to more problems. So potentially sleeping more might help delay the onsets of Alzheimer's disease, because it's thought that you are clearing out all the buildup of not just toxins, but things in your brain you want to get rid of. That happens during sleep. So if we're sleeping less, we're causing trouble for ourselves down the line. I can say personally, there's always a part of me that feels like going to sleep is losing time, that I could be, watching a movie or doing some work. And what am I doing? 8 hours, I can sleep in 6 hours and I can have 2 more hours of living. But if there are consequences down the line, we are stealing from the future by not sleeping.
Well said. I think that was very eloquent. So, but you're not concerned that by manipulating the memory replay and sleep, we're brainwashing. What you're saying is the effects of that are really quite subtle compared to things like what the Tonegawa lab are doing in wake. So right. That's reassuring. So yeah, I guess just one final question. What are your, answer as you feel comfortable, but what are you doing right now? What are the exciting things that you're asking in your lab at the moment?
Yeah, so we're sort of looking at 4 things. So the first is, we know that replay reflects recent memories, but we don't know how long something replays for. So if you come back to the study I said, where you sleep deprive someone for 2 days and then the bias goes away, that suggests that there is something time limited. So is it that replay stops after that time automatically if you didn't replay it, or does the bias go away? We need to have some measure of how long a memory gets replayed for. And that's difficult to do technically, because when you're recording from neurons, you assume that there's some physical movement of the electrodes relative to the brain over time. And so typically you can record maybe 6 hours, but not over 2 days. So we have some ways to work around that to see if we can get a measure of what is being replayed in the brain. The second thing is that when replay occurs, we speculated that an important memory is going to be replayed more. And that's been looked at a little bit, but we wanted to explore. So for instance, reward on the same track, if the animal can go right and get a reward and go left and not get a reward, will it replay the right more than the left? We're taking a different approach where the animal has different environments it's exploring, and one environment is more rewarded than another. Alternatively, you can also look at the amount of experience the animal had or how recent that experience. So we're trying to manipulate all these different factors to see what causes more replay. And likewise, when you have more replay of something, what is the dynamics of it? So you have more replay and it's at a constant level, or does it peak early and then go down so that other memories can reactivate. So it's essentially one question, but with many, many avenues to explore. The last thing that we're looking in the lab is this idea of a memory engram, an inhibitory engram. So when you have replay, you are presumably causing neurons to wire together, to connect together, so that the sequence of how the neurons should fire in cortex is stored in some way. Now, presumably, if you're only exciting the neuron, then it would be able to spontaneously reactivate, and that's bad, right? You would have an hallucination. And maybe that's what happens in schizophrenia, that you have too much excitation and it's like the brakes don't work because you've just pressed on the accelerator so much in your car. So the thought is that with excitation, you should also have inhibition. So that's called an inhibitory engram. So you have both excitation and inhibition that scale up together. The advantage of this is that if you, at a later time, want to remember the memory, you could decrease the inhibition and then the excitation comes out and the memory expresses itself. But the memory itself would not impair the neural circuit from normal perception because the excitation and inhibition are balanced. So we're exploring to see, do these inhibitory engrams exist in cortex and how they would manifest. That's still early days for that experiment.
Wow, that's quite a handful. It sounds like you're going to be busy for a long time with these three different projects, which is great. I mean, it's what you want as a scientist. All right, well, fantastic. I mean, this has been really interesting and really fun, and I really appreciate your eloquence and your time.
Thanks, Penny. It's been a pleasure.
You've been listening to the Sleep Science Podcast with me, Penny Lewis. My guest today was Dr. Daniel Bendor from University College London, and our producer was Sophie Smith. If you've enjoyed this show, please consider subscribing to the podcast or liking us on Twitter. We're planning a Q&A session for the last episode of this series, so if you have questions about this episode or anything else sleep related, please send them to us on Twitter at #SleepSciencePodcastQ&A. Thanks for listening, and until next time, sleep well.